The overall goal of this procedure is to prepare adult Drosophila eyes for thin sectioning, allowing for the detailed assessment of mutant eye phenotypes such as planar cell polarity defects. This is accomplished by first cutting off the heads of anesthetized flies. The eye tissue is then fixed and embedded in an epoxy resin. Once the resin is solidified, the embedded eyes are trimmed for sectioning. The final step is to thinly section the eyes using a microtome equipped with a diamond knife. Ultimately, analysis of the eye section will allow the characterization of eye phenotypes of novel mutants or the quantification of genetic interactions between genes of a signaling pathway, such as the PCP pathway. Both will help to characterize the physiological role of newly identified genes.
This method can help answer key questions in the Drosophila field, such as how novel mutations affect eye development or how genes interplay in genetic interaction assays. Several steps of the procedure of eye embedding, such as aligning the eyes in sectioning molds, are way easier to understand visually rather than in text form.
To begin, put on a pair of gloves in preparation for the fixation of the fly heads in glutaraldehyde and osmium, both of which are toxic chemicals. Next, anesthetize flies using CO2 and tap them out onto a fly pad protected by Whatman paper under a dissecting microscope. Typically, sort out 6 flies plus an additional fly to ensure 6 embedded fly heads per genotype. Hold and gently press the fly's thorax with tweezers and then remove the fly's head using a scalpel. Stabilize the fly head.